Hi students, today we are going to go over how to complete the 731 Exploring Living Things. What structures do all living things have? If you remember 2 lessons ago, we talked about caterpillar soup and you got to learn what caterpillar soup was, and then you got to decide whether or not caterpillar soup was living or not living. You just told me what you thought. We didn't have a whole lot of information about caterpillar soup at the time, but you just told me what you thought if it was living or not living. And then yesterday, you had a chance to look at what functions living things have, what living things do. So we learned that living things can grow, they can reproduce, uh, they can they have to consume energy and they produce waste. So if you remember right, we talked about or you learned about all of those functions that living things do. So today what we're going to focus on is what structures do all living things have. So we'll go ahead and get started. Remember to pause the video when you need to complete certain parts of this activity, and then you can restart the video once you have completed the parts. So on slide 2, it just reviews the functions of living things that we talked about yesterday. So we talked about how all living things perform the same basic functions. So if you want to know if something's living, you can see if they eat or take in matter, poop or get rid of waste, if they grow and develop, they reproduce, and if they respond to the environment. So if they do perform all of these basic functions, then we know that something is alive. So remember, I bolded these basic functions. It's really important that we remember these basic functions of living things because these functions help us determine if something is living. We can also look at specific structures of living things to determine if something is living, which we are going to talk about today. But before we get into structures, if you would like to, you can watch some of the basic functions that living things perform in specific organisms. So I've attached some videos here. You can watch a plant grow and develop. You can hear prairie dogs respond to danger in their environment. You can watch and maybe hear a sea cucumber getting rid of waste. And then if you want to see some more organisms with waste removal, you can also watch these. These videos are not required. This is in addition if you want to go back and look at some of these organisms performing basic functions that living things perform. So we're going to go ahead and get started with structures. So what about the structures of living things? If you look at these 4 living things, we've got slime mold, we've got an elephant, this is a foxglove plant, and then this is an oyster mushroom. Do you see any patterns in the structures of these organisms? So do you see anything as far as structure, the way that they are built? Do you see any similarities between these? So think about that question for just a minute. We are going to go to the next slide and talk a little bit about scale. We've talked about scale before, but I put an activity in here to help us review scale so that you can understand the activity that we're going to be doing today. So while we are looking at the scale activity on the next slide, just think about this question. As we look at living things, do we see any patterns in the way that they are built? So let's go ahead and move on to this activity that helps us review scale and structure. Now, it's difficult to find patterns in the structures of living things if you look on the surface. So if you look at the pictures from the previous slide, slime mold, elephant, foxglove, and oyster mushroom, it's really hard to find patterns in the structures that connect them or that make them similar. So we know that they're living because they perform the basic structures or the basic functions of living things that we talked about yesterday, but their structures are very different. But what would happen if we observed the structures of a living organism from a different scale? So here we're looking at these organisms from a large scale, but what would happen if we maybe shrunk the scale down a little bit? Would we find any patterns? So we're going to take a minute to review the idea of scale and play around with a couple of scale interactives. So I want you to spend 5 minutes on this particular slide, exploring the Scale Interactives. If you click on these links here, they will take you to two Scale Interactive websites. This is what the first one looks like. You can see the scale of different things. This is the size of a coffee bean compared to a grain of rice, compared to a sesame seed. And then as you zoom in, you can see things get smaller and smaller, and you can compare the scale of different things as we get smaller and smaller. And then you can do the same thing with this uh, interactive called the Scale of the Universe. I prefer this one better to the other one. So here we are at a basketball, the largest hailstone, Russell's teapot, 1-inch ruler. So we're looking at items that are about 1 meter. If you zoom out, things get bigger. If you zoom in, things get smaller. So you can kind of explore these two websites, and they can help you see the different scale of different objects. So then what I want you to spend time doing after you are exploring, um, after you are done exploring, you've got a bunch of pictures here. You're going to arrange them in order from the biggest scale to the smallest scale object. Okay, so you'll just start um, with these objects. You'll have to click on the pictures to move them around, and you'll decide which of these are the smallest objects and which ones are the largest, and then just arrange them in order on this line from biggest to smallest. So again, I want you to take no more than 5 minutes exploring the scale interactives and then arranging these items on the timeline. When you're done with that, after 5 minutes, go ahead and start the video again. All right, so now that you have reviewed the idea of scale, what we are going to do today is take a look at living things on a different scale. We're going to look at a very smaller scale than looking at the big scale, which is what we did on slide number 2. Slide number 2 got a big picture of what these living organisms look like. We're going to shrink our scale down using a microscope. So we're going to investigate what the structures of living things look like on a microscopic scale. Hopefully we'll be able to find better patterns when we look at a microscopic scale, and then these patterns we can use to help us figure out if our phenomenon from the first lesson, caterpillar soup, uh, we can see if those patterns will help us be able to tell if uh, the caterpillar soup is living or not. So we're going to be using this microscope. It's a virtual microscope, which is kind of nice because we're not in the classroom, so we can still see what a microscope would help us be able to see by using this interactive. So I'm going to do the first one, the onion sample with you, and then you can do the bone and the cheek cell. This should only help. This should only take you about 15 minutes to complete. So make sure that you're not spending too much time looking through the microscope. You just get the information you need to complete the slide and then you move on. So I'm going to show you how to do this interactive, and you can work along with me as we do the onion cell. So when you click on the interactive, that's this link right here. If you click on the link, it will open up the interactive microscope, and you click launch interactive. And it's going to give you instructions for how to prepare a slide. Now, if you look, our table says to prepare the onion slide first. So it says to view a specimen under the microscope, click the slide box to select a slide. So we click the slide box, it gives us a slide. Now we get to choose a specimen and drag it onto the middle of the slide. So we're going to start with the onion. So we're going to grab the onion and put it on the middle of the slide, and that's going to give us a specimen on the slide. So you choose to view the onion cells. To see the onion cells, they have to be processed. So then we have to click the processing equipment on the shelf and drag the dropper to the specimen. So the processing equipment is here on the shelf. So we're going to click on it, maybe, and then we're going to drag the dropper to the onion cell. And you have to place it just right in order to get the dropper to drop. Okay, there we go. So now we're going to click on the box of cover slips to cover the specimen, and then it will be ready to view in the microscope. So then it gives you information about light microscopes and how much the magnification is. By itself, this lens piece right here, the ocular eyepiece, magnifies 10 times. And then down here, there are 3 objectives that magnify it even more. The low power objective is 4 times, so then if you go 10 times 4, that's 40 times the magnification. And then the medium power is 10 times, so 10 times 10 is 100 times. And then 40 times is the higher power, so 40 times 10 with the ocular eyepiece is 400 times. Okay. So then this is explaining how we get the total magnification, and then we can rotate the objectives to get a higher magnification. Okay, I think we are ready to look at the microscope. All right, so we need to bring this into focus. We can see an image in here, but it's not focused. So we're going to focus first with the coarse objective and then with the fine objective, or the fine focus, sorry. The objectives are here. The focus knobs are on the side. So we can see that this is what the slice of onion looks like at the lowest magnification. Down here in the description, it says that you can see small rectangles that are lined up side by side. Those rectangles are what we call cells. And you notice a darker dot inside each of the smaller rectangles. That's the nucleus. And we're going to switch to the higher magnification. So let's see, how do we do that? I forget. Okay, so we have to click enlarge on the microscope here, and then that will allow us to switch to a higher magnification. So now we're at the 10 times. We're going to do the coarse focus and then the fine focus, and now we can see those cells lined up side by side a little bit clearer. But we want to get to the highest magnification to get the best view of those onion cells. All right, so now you get a pretty clear picture. You can see those small rectangles that have a dark outline, and there's a bunch of them lined up side by side. These are onion cells, and inside the onion cells we have a nucleus in each cell. That's these purple blobs. So what we know is that when you shrink an onion cell, when we get a closer, smaller uh, picture of a piece of an onion, we get onion cells. So we can see onion cells microscopically. So remember, we are going to look for um, patterns to see if living things have the same sorts of things at a smaller scale or microscopic scale. So we're going to go back to our table on slide number 4, and we are going to, what I'm going to put is, we could see the best image of the onion at 40 times the magnification, so I'm going to do 40x. And I'm going to describe the structures that I observed. So I saw small box-like structures that had a dark outline and a purple circle inside. The simulator called the box-like structure cells, and this purple circles nucleus. Okay, so we're going to go back and we're going to do the bone. So after you've cleaned up, you can go and prepare a slide. Remember to follow the instructions down here. It will tell you what to do piece by piece. This time you're going to look at the bone cells. And remember, we're looking for patterns to see if there are similar things at a microscopic level that all living things have in common, similar structures. So I would advise you, where it says magnification here, I would advise you to look at all three magnifications, 4 times, 10 times, and 40 times, and then just find the clearest picture. And whatever magnification is the clearest picture that you can get the most information, I would put that here in the table and then describe what you saw at that magnification. And then when you're done, you're going to look at your samples and what you've written for each sample for your onion, your bone, and your cheek, and describe any patterns. Do you see any patterns as far as structure goes? Do they have similar structures? What patterns do you observe? So again, this should only be 15 minutes total, and when you're done, you can go ahead and restart the video, and we'll move on to the next activity. All right. So you should be done with slide number 4. Your table should be completed, and you should have some patterns. Many of you probably noticed that one of the patterns that we observed in all three samples is that they all have little tiny structures that we can't see without a microscope. When we use a microscope, they're visible, but we can't see them without a microscope. So we have to look at a smaller scale to be able to visualize these smaller structures. These structures are called cells. Now, I've given you some pictures of all three of those different things that we looked at, the cheek cells, the onion cells, and the bone cells. And you can see that there are arrows pointing to individual cells from each sample. So all of these pink blobs here are cheek cells, and then you can see the arrows are pointing to individual cheek cells. The onion cells are also side by side connected to each other, but these arrows are pointing to individual cells. And the bone cells are a little bit harder to see. These dark spots are they're nuclei, which is plural for nucleus. And you can see that they're kind of similar to the nuclei that you see in the onion cells. But these arrows, the red arrows, are pointing to individual bone cells. So the biggest pattern that we observe as we look through the microscope is that living things are made of small structures called cells that are only visible through a microscope. So what we're going to do next is on slide 6, I've got some other pictures of cells, or of different types of living things, and there are cells in each one of these slides. So these are microscope slides, just like we did with the virtual microscope. And we've got a liver slide, we've got potato slide, intestine, wood, leaf, and plant root. What I want you to do is practice identifying where the cells are. So what you're going to do is you're going to go up here to your drawing tool, and you're going to, or your line tool, sorry, that's your line tool, and then down at the bottom there's a scribble tool. And you're going to practice outlining the cells in each of these slides. I've done the liver one for you. As you can see, there are several liver cells that I have highlighted in yellow. I will go ahead and do the potato one. on this video so that you can see another example. So I remember from the virtual microscope that these cells sometimes have a really dark outline, which makes them easy to identify. So I'm thinking that that part that I just outlined there would be a cell. So I'm going to highlight that in yellow and maybe make the line a little bit heavier so it's visible. And I'm going to grab my scribble tool again, and I'm going to try to outline a couple of others. This one looks like it's another potato cell right here. Okay, and then I'll change the color and the weight so that it shows up a little bit better. Okay, and then let's do another one. There are lots of potato cells in this slide, but I'm going to just do 3. So what I want you to do with all of these samples is practice identifying where the cells are in each sample. So just a little bit of a hint about a couple of these. The leaf and the plant root should be pretty easy to see, but the intestine and the wood, you're going to have to increase the magnification a little bit more to see individual cells. So if you look, I've increased the magnification, and now you can see where the individual cells are lined up next to each other. And if you go down to the wood, the same thing. You can see the individual cells with the dark outline lined up next to each other. So if you want to zoom in to be able to highlight the intestine and the wood, then you can do that. You only need to spend about 5 minutes outlining some of the cells on the sides of this page. So take 5 minutes doing that, and then you are ready to move on to the next slide. All right, on slide 7, I have some of the cells outlined for each of the samples. So if you want to go back and check your work from slide 6 and see if you were right and you could identify correctly the cells on each of these samples. If you notice, like I said with the intestine and the wood, the cells are even smaller in our visual field than in the leaf or even the liver or the potato. So you had to magnify it even more to be able to see the individual cells and circle them. All right, let's move on to the next activity. All right, so what are the structures that all living things have in common? What did we see in the microscope when we looked at the onion, the bone, and the cheek? What would we see if we were to take samples of all living things? What did we see in the liver, the potato, the intestine, the wood, the leaf, the plant root? We see that all living things have cells. So that is what I want you to get from this lesson, is that even though structures we can observe with our eyes seem very, very different when we looked at all of these organisms from slide 2, the slime mold, the elephant, the foxglove, and the oyster mushroom, they all look really different structure-wise. But when we look at them on a microscopic level, we can see that they all have one thing in common, and that is that they have cells. So now you know some of the characteristics that living things share. Summarize what you know, what you've learned yesterday, and what you've learned today in the boxes below. So you might have to go back to yesterday's lesson to do the first part, but what I want you to do is take 5 minutes, no more than 5 minutes, to summarize what functions living things share. You probably don't even have to go back to yesterday's lesson because I've included those functions here on slide 2. If you remember, they're bolded. So you could use those in your summary here. And then also, we just finished talking about what structures all living things share. So this should be really, really easy for you to fill out. So like I said, take only 5 minutes to finish this slide and then move on to the next slide. Okay, now that you have summarized what you have learned about the functions of living things, and what structures all living things have in common, we are going to go back to our phenomenon from the very first lesson that we had in this unit, which was the caterpillar soup. Do you remember the caterpillar soup? Do you remember that the caterpillar soup is kind of an intermediate stage between a caterpillar and a butterfly or a moth? So we were trying to figure out if caterpillar soup is considered living or not. So what we're going to do is we're going to kind of summarize what we know about each stage in the development of a butterfly or a moth, um, what we know and what we still need to try to figure out. So I've got this table to kind of help us figure out what we know and what we still need to figure out. And I think I'm going to do this along with you. It should not take longer than about 10 minutes, but I want to make sure that we really understand what it is that we know about each stage during the metamorphosis of a caterpillar into a butterfly or a moth. So what we're going to do is we are going to look at each stage. We've got it listed here at the top: caterpillar, caterpillar soup, that's the in between, and then the butterfly or moth is what the caterpillar metamorphosizes or changes into. So what we're going to do is we're going to look at everything that we know about living things. We discovered today that living things are made up of microscopic structures called cells. And then yesterday we learned that living things respond to the environment. They eat and take in matter, they poop and generate waste, and they also grow and develop. So what we're going to do is we're going to decide if each of these, if the caterpillar soup, and the butterfly or the moth, what we're going to decide if we can give them a check mark. Yes, they have this or they do this. If we know for sure that it doesn't, then we're going to put an X. But I've also given you an option to put if you don't know. If you don't know for sure. So if you don't know, then put a question mark. So I'm going to do, uh, I'll do the first one with you. Okay. So the caterpillar, do we know that it's made out of cells? Well, we can assume that it's made out of cells because we just learned today that all living things are made of cells. So I'm going to put a check mark there because I think that because caterpillars are living, that they must be made of cells. Now, if you don't know for sure, you could put a question mark there, and then maybe, maybe we could figure out a way to investigate to see if caterpillars are made up of cells or not. Do they respond to the environment? So, well, let's go back to cells. In the cell box, I'm going to put, uh, I know that caterpillars are living, and I know all living things. So I will assume that that caterpillars are made of cells. Okay. And then let's go to the next one over, caterpillar soup. Um, I don't know if it's living, so I don't know if it's made up of cells. So I'm going to put a question mark there. And then with the butterfly and the moth, I know that they are living, so again, I'm going to assume that they are made up of cells. So I'm going to add that to my notes here. I know that caterpillars and butterflies or moths, that they, I'm going to change caterpillar to they. And then I'm going to add, I don't know if caterpillar soup is living, so I don't know if if there are cells in caterpillar soup. Okay, so that's a pretty good explanation for the first box. So then what you're going to do is you are going to go through each of the four functions of living things and decide if the caterpillar soup, or the butterfly or moth perform each of these functions. Now again, if you know for sure, you put a check mark. If you know for sure that they do not, you'll put an X. If you don't know, then put a question mark. And then in the notes section, explain your thinking. So again, you have about 10 minutes to complete this, and then you can push the pause button while you are doing that. And then when you're done, go ahead and push start again, and we will do the last activity. All right, so hopefully you filled out your table on slide number 9. We are actually going to be using that same table in tomorrow's lesson, so make sure you get that filled out. We are now on the last learning activity. So let's take a minute to think about our phenomenon, which is the caterpillar soup. So we know the caterpillars are living, we know that the result of the chrysalis is a butterfly or moth and they are living, but our question is still uh, if the caterpillar soup, which is the intermediate stage, if that is living or not. So let's consider what we know and what we don't know about the caterpillar soup. Um, we did that, we summarized that on the previous table. So if we have a question mark, then we don't know. If we have a green X, then we do know. A green X or a green check mark or a red X will tell us that we do know for certain whether it does or does not. A question mark tells us that we still don't know. So what we're going to do is I want you to, in one sentence, tell me what additional information would help us be able to figure out if caterpillar soup is living. And then how will this information help you? I've given you a sentence frame that's highlighted here in yellow. Please use the sentence frame to be able to write your answer here. You can write more than one thing that we need to figure out about it. So here's the sentence frame. We need to know if caterpillar soup blank. So what do we need to know about the caterpillar soup? And then this will help us figure out if the caterpillar soup is living because, and then you tell me why it would help us figure out or how it would help us figure out if caterpillar soup is living or not. So you should only need to take about 5 minutes to finish this slide, and then you are done. If you have any questions, please remember that I'm available through email. You can send me a message on Canvas. You can also let me know if you are planning to join me during office hours, and I will be available again through email, through Canvas, and then even you can join Zoom if you would like. So let me know if you have any questions, and good luck on completing today's learning activity.